Can you all see my screen? Do you see all my, do you see
Yes,
screen?
sir.
Okay. So in the tools to study development, today we are going to learn very, very important tools which have been proven to be extremely successful in understanding various processes in cellular function, but also in particular the development. These are not dedicated only to development, but they have been used successfully. For example, whenever we think of a developmental process, whether it is early embryogenesis or let's say the processes in gastrulation, processes in neurulation, processes in organogenesis, you know, morphogenesis in tissues and cell organization, etc., in pattern formation. We in cell and molecular biology, we think that we have to discover gene network or protein networks which are responsible for development of a specific pattern in a given tissue or an organ or an organism. Now, classically, we perform genetic screens. What we simply do, we use EMS as a mutagen. So we choose a mutagen, or in flies, for example, we can also use P element. In Arabidopsis, they use tDNA insertional mutagenesis using agrobacterium. So this is a transposon. This is a tumor-inducing plasmid of a bacteria. These have been engineered in such a way that when they land into the genome, wherever they are going to land into the genome, they are going to mutagenize the gene. So for example, if there was a gene here in the genome on a chromosome, so this in
Sir,
yeah.
sir, we are not seeing the slides. We are just seeing some folder open over here. So the slides are not visible.
Oh, come. So why others are not telling me?
Sir, I just realized the same.
Okay, let me go back. And okay, I open again the file and so do you see my screen now? Do you see the?
Yes, sir. Now the slides are visible.
Now when I go into PowerPoint mode, do you see it?
Yes.
Do you see what I have written? P element, tDNA, etc.
Yes, sir. The slides are visible.
Okay, good. So I was telling you that you can use transposon mediated mutagenesis in fruit flies or tDNA mediated mutagenesis in Arabidopsis or plants. Historically, conventionally, when such engineered mutagens were not there, DNA-based mutagens were not there, people were using EMS mutagenesis, which is an alkylating agent. All the idea was to, cause random mutagenesis if these are different chromosomes of fruit fly, for example, 4 chromosomes. So any of the mutagenesis approach you use, you are going to cause random mutagenesis in the genome. Each line I am drawing on the chromosome is indicating independent mutation event, which means an individual fly. Last time we did saw how they set up a genetic screen. Now, before you set up a genetic screen, you have to be very smart and think of your screening strategy. How are you going to screen your mutants? Because if you expose 10,000 males to a mutagen, there will be a huge number of mutants. Now, you have to see which ones are mutants of your interest, which means you are basically going to predict a phenotype. You are going to have a phenotype. If you are interested in eye development, you will think of, this is the wild type eye. So if I'm working in some kind of looking for oncogenes or tumor suppressors, I have to see what kind of phenotype it will be there. If I'm looking for eye development, then I will exclude, let's say, flies which may show thoracic or wing phenotypes, because I'm interested in development of eye. Similarly, if you're interested in wing development, you will be focusing or looking at phenotypes in the wing and ignoring what is happening in the eye. You can also design reporters in transgenic flies, which is a more advanced strategy. So all this is about how you are going to select your mutant of interest. And once you have those mutants, you can then go and map the mutation. After mapping, you find the gene, and then what we call characterize the gene. Characterize seems a very small word, but this word actually means you have to discover each and every function of this gene, how it performs its function. Okay. Now, you can also think of doing a genetic screen in which you look for modifiers of the existing mutation, which means, let's say there is a mutation which gave you such an eye phenotype. Okay, wild type eye in fruit fly look like this. Or there is a mutation, and if you have a mouse, let's say this mouse has defects in limb development. Limbs are not developing properly. And you know the gene, you already know that particular gene, that particular mutant. You can use this mutant and then look for suppressors or enhancers of this particular mutation. And we call them modifiers of the existing mutation. That will tell you the gene X in the mouse, which is involved in limb development. Mutation of X causes limb development. But now when I use this, and this mouse is mutagenized again, if I modify limb development, either the phenotype becomes extremely severe or it goes back to normal. It means I am discovering new genes which are interacting with the gene X. Okay, and normally the purpose of performing such mutagenesis and genetic screens, the purpose is to kill the gene function, find loss of function. It's written, the right word in genetics is loss of function mutation. Due to genetic intervention, the function of the gene is lost. And as a result, we see a specific phenotype. This is one approach. Then let's look at some molecular experiments. In the molecular experiments are actually the ones which can be done on the mutants you have discovered already. For example, it says specific expression pattern. You have your mutant and you have wild type. Now, in order to understand and characterize the function of gene X we have discovered, we can simply do Cdna subtraction, or what we also call the differential display. The purpose of Cdna subtraction or differential display is actually see which genes are, the expression of which genes is altered. In wild type, let's say, you take limb birds, and what you see from A to Z, all genes. Normally in the limb birds, let's say A is on, B is on, C is on, D off, E off, F is on, G, H, everything. So you have a pattern. Now, what you're going to do, you're going to look at the expression of mutant and see A to Z, what are the alterations. Now, let's say you find in the mutants, A is off, B is off, M is off, N is off, but K was off here, K is on there now. Other genes, some of the genes. So you have found, let's say, 50 genes going down, 50 genes, or let's say 150 going up. Now it does not mean your mutation is impacting these genes directly, but you are in a position where you can deduce that my gene of interest out of all these total 15,000 or 20,000 genes, my gene of interest, which is when it is mutant, it's impacting these pathways. You will look at these genes, these 150 and 50, and then you will try to see, okay, is A is interacting with K, with M is talking to A, okay? If there is a circuit, how many different circuits are there? Genetic circuits or genetic networks are there? You can also do, so this is to monitor such expressions, you can do Cdna subtraction. There's another technique called differential display, or you can simply do, this can be achieved through microarray. And nowadays, instead of microarrays, we do RNA-seq. You take total RNA of wild type, total RNA of mutant, and you do next generation sequencing. All the RNA which is expressed, let's say I try to draw here, this is wild type, this is mutant. Wild type, your mutant. All the genes on different chromosomes which are on or off, you will see every possible gene in wild type, their expression pattern. Then in the mutants, you will see where you see a dip and where you see more expression pattern. The whole idea is actually to find, and you can do this characterizing your mutant, or you can do this through already existing mutants. Mutant is already there. You haven't done the screen, someone else did it. And you want to see while characterizing function of that gene, because that mutant phenotype is phenotype of interest to you. Okay. Now, in order to determine the expression pattern, like here we said limb specific expression pattern or eye specific expression pattern. In a more classical approach, which was established in flies and then also adapted in Arabidopsis and other systems, is enhancer and promoter trap screens. Let's say we are interested in eye development. Okay. We are interested in eye development. And we want to discover all the possible genes which contribute to development of eye. This is the adult eye. In fruit flies, the earliest eye cells are somewhere here, few cells. From here, all the way to there, what are the genes which contribute to eye development? It was a very intelligent screen. Whenever I think about this screen, I can't help appreciating people who designed this screen. What this did, they said, okay, any gene which is only expressed in eye must be regulated by some tissue specific elements of that gene. We know the definition of gene. We know there are tissue specific enhancers for different genes. So if there is an eye specific gene, it means that gene has some, from here that gene has some regulatory elements, cis regulatory elements, which is helping expression of this particular gene only in eye, and it's not expressed in wing or legs or etc. It's off. So what they did a screen, and this is called enhancer trap screen. Okay, do you still see my screen?
Yes, sir.
Now, what is enhancer trap or promoter trap screen? So we know normally a gene has its own promoter, but in the enhancer trap, what they did, they designed a specialized reporter system having, let's say, bacterial LAGZ gene. And in front of this, they just had what we call minimal promoter. Now, minimal promoter means maybe just a Tata box. This minimal promoter cannot activate LAGZ at all, at its own. And then they had, let's say, white as a transformation marker to select the transgenic flags. What they did, they injected this, and this was packaged in a P element vector. They injected the pole cells, they prepared the transgenic flies, and in the G0, you don't find any red eyes. In G1, you find the red eyes, which means you have white 1118 is a specialized genotype in which eyes are white. So when we have transformation marker, it means we have to use a fly which has eye color, which is mutant for eye color. Now in G1, you find your transgene is there. What we are going to do now, we generated, let's say, 5,000 such transgenic flies. What is the idea? The idea is that in these transgenic flies, wherever we will have this particular construct, which is referred to as enhancer trap construct, if it will land this construct, which is LAGZ and then the minimal promoter, if it has landed near a gene next to an enhancer, which is actually this enhancer in wild type is acting on promoter of the gene and then activating eye specific gene. Now, since eye enhancer has landed there, enhancer trap construct, now this enhancer will act on the minimal promoter, which is in our construct, and will activate LAGZ. What we will do, we will take all these 5,000 flies, we will dissect, and we will dissect their eye imaginal disc. Okay, 5,000 independent mutants. We will stain for LAGZ activity by adding XGAL, which is a substrate of the LAGZ gene, and then see, and as a control for each fly, we also isolate their imaginal disc, Halter disc, the wing discs for each of this 5,000. And then try to see, let's say we find very characteristic expression of LAGZ here, but in neither leg, wing, Halter, etc. In another one, we do find leg Z expression in I, but is also present in wing, but not in leg and Halter. So we will not be interested in this one, but since we are interested in I specific, we will go and chase this particular gene. Now it's very easy since we know where this gene has landed and we know the P element sequence, DNA sequence of our P element on these inverted repeats. We simply design primers and perform inverse PCR. Go back and read about inverse PCR. I think I have explained this. By doing inverse PCR, you simply have a primer here. And since these are inverted repeats, this primer, single primer, will bind there as well. It will go like this and like this, and we add random hexamers, which is the other primer, and then we have amplification through PCR from single fly PCR, and we get PCR products. We sequence them, and then we can tell where exactly our transgene has landed. In this particular fly, a gene A is mutated, and that is responsible for eye development. In order to prove that it is eye specific, in order to prove that we have found a eye specific gene enhancer, we clone this enhancer from the wild type. And what we do, we have now this gene A enhancer and tag it with GFP or LAGZ or whichever reporter. And then we make transgenic flies again, inject them, and see in the transgenic if I see only eye-specific expression. Take imaginal disk, wing disk, and see where the expression is. If we determine that it is like this, it's a big discovery for us. So people have done these enhancer trap screens, and through these enhancer trap screens, they have cloned many eye-specific, wing-specific genes. Same is the case for, so that was an enhancer trap. If I say promoter trap, it's same. In that case, you're just going to have leg Z gene, then mini wide transformation marker, no promoter, no enhancer, here is nothing. This P element will be injected in flies. In the transgenic flies, idea is when it lands in a gene whose promoter is next to our gene, the gene promoter is going to activate our leg Z, and rest is the same. So what you have done, you have found regulatory elements for tissue specific genes. Not only regulatory elements for tissue specific genes, you have discovered and you have cataloged eye specific genes. Is it clear so far?
Yes, sir.
Okay. Now, these were different approaches. Yeah. Now, there are other things as well. So this was starting from scratch. This was, either it can be your own mutant or some other known mutants, and you have tried to determine gene expression, characterize those, or you are interested in tissue specific genes, and through enhancer or promoter trap screens, you have found new genes. All these are different ways, different approaches to start a new scientific question, bring answer to a new scientific question. Another approach can be, you can simply start with a homology to a known gene. What you have is, you have let's say we have a known mutation in UBX, we know its phenotype, we know it's the bithorax phenotype, etc. Now, what we found that we have a homologue in humans or mouse, or let's say we take a new organism, zebrafish. Now you know the function of UBX in flies, but you don't know whether it will behave similarly in zebrafish. This organism is invertebrate, whereas zebrafish is a vertebrate. So you simply, based on the homology, you say, okay, I take zebrafish UBX gene. I'm going to start a project and I'm going to go through all these different things. Nowadays, when the functional genomic tools and the, compared to 20 years ago, nowadays we have ready-made mutants already. When we discover a new gene in fruit fly, the first thing we do, we go on the fly base, which is the genome, a web page which catalogs every possible mutation in fly. We go on the fly base and try to see if we have already available mutant. With the presence of, in the presence of CRISPR or RNAi, people have already made and they are commercially available for $10 or $25. Instead of you going through pain of 2 years to generate new mutant, you already have a mutant available. So if you have a mutation for zebrafish UBX, you can just go and characterize this using all these approaches, and then try to see if the UBX actually plays a role in the pattern formation and tissue and cell specific specialization in zebrafish too. Now, we also, in my last lecture, I talked about ectopic expression. Expression of UBX leads to development of haltear in flies. If you have UBX on, it's haltear. If you have UBX off, it's wing in flies, fruit flies. Now, in order to understand that UBX is very critical for developmental processes, you do ectopic expression. We did this in our last lecture. What we did, we said, let's take eyeless enhancer, which means expresses only in the eye, clone UBX under it, and then express in the adult eyes of fly. Or I take wingless enhancer. This is enhancer for wings, the wingless pathway. I clone UBX. Now what I am doing, normally UBX is off in eyes, so I'm ectopically expressing. I'm expressing UBX away from its natural location, which is in the haltear. If I use, let's say, wing specific expression, now what will happen? Normally in the wild type wing, if you take the wing imaginal disc, which result in development of adult wing, you don't find UBX expression in the wing pouch, etc. Now, if you ectopically expressed UBX in the wing imaginal disc, this will be converted into a haltear, a small structure. Okay. Is it clear so far? आप लोग सारे हो भी कि नहीं उधर, या मैं ही शाम में बोल रहा हूँ? Are you all there?
Yes, sir, it's clear.
Yes, sir.
So when we discover a gene and a mutation. Of course, in case of development, we try to see whether a newly discovered gene is maternal effect gene or is it a zygotically expressed gene. If it is maternally expressed gene, of course, by simply doing in situ hybridization at early embryonic stage, which means in unfertilized, we can try to see if RNA, we do find our gene of interest through in situ hybridization in the unfertilized eggs. It means mother has deposited this. Then we also try to see if it's lethal. If it is lethal, lethal means its mutation in this gene is causing death. It means this is essential gene, extremely essential gene. If it is lethal, then we have to see where in developmental time, let's say this is day 0 and this is day 10, this is 24 hours, this is still egg, this is 48 hours, first install larvae, 72 hours, this is second install larvae, etc. This is fly. It can be C. elegans, life cycle 0 to whatever, I don't know the exact days, the hours for C. elegans. Let's say this is the adult. 0 is the time point 0 for life development, and this is the adults. So you have to discover the phase of development, the time of development, where this lethality is happening due to loss of your mutation. It may grow all the way to pupal stage, okay, up to 6 days. And then from the pupa, it never hatches. It means it's living like normal larval stage, but it's pupal lethal. It could be embryonic lethal, which means you never see larva development. So you have to define then the phase where this death is taking place. Then, of course, you characterize your mutation. And in classical genetics, we have like loss of function mutation, we have gain of function mutation, and then loss of function mutation is again, there are two kinds. Amorph means, A means absence, null. So total loss of function. So loss of function can be completely absent gene. Due to mutation, gene has lost its function. And then the other is hypomorph. Hypo means reduced, less. Gene is there, but it's weaker and partial loss of function. Maybe it's still able to do 50% of its, 25% of its function, but gene is there, okay? Hypomorph are usually weak mutations, and amorphs or null mutations, these are very strong mutations in loss of function. The other mutations can be gain of function mutations. Gain of functions are normally dominant mutation, and loss of function are normally recessive mutation. Gain of function can be antimorph, as the word says, gain of function. Due to mutation, your gene of interest has acquired some new function, which is opposite to the effect of wild type. So in this case, this is antimorph, opposite of wild type. Neomorph means new function. Neo means new. So it has acquired a new character, a new function. And hypermorph means, hyper means it has strong effect than the wild type. Go back and brush up your genetics lecture 221. There you find how we develop, how we conclude your mutation is amorph, hypomorph, antimorph, neomorph, and hypermorph. This is, consider this your assignment. How do you conclude a given mutation is amorph, hypomorph, antimorph, neomorph, or hypomorph. You need to show us. You can draw figures, you can write lines, but you have to draw pictures to show how to give explanation. Okay. And then finally, as we already said, loss of function mutations are normally recessive and gain of function are dominant. There's not really 100%, but you have to characterize by crossing with wild type to discover whether your mutation is recessive or dominant. As we said, mutations can be lethal. It can be embryonic lethal. When we lose function of a gene, and if it turns out to be lethal, can we still study this function or this process, or we cannot? For example, if you're interested in mitosis or cell cycle, okay, let's say G1, G1S, G2M. Let's say you're interested in the cell cycle, and you do conventional mutagenesis. You will never be able to find a gene, because your mutant is any gene which will be playing a role in cell cycle. It will be dead. This mutant will not divide mitotically. When it will not grow, you cannot study this particular process. Or in developmental stages, if you're interested in early stage development and your genes are important, these early stage embryos will simply die. You will never be able to study early stage development. So scientists came up with a strategy to induce temperature sensitive mutations. And there is a way, very technical way, to generate mutations in organisms which are temperature sensitive. What will happen in this case? Now you have induced mutation. The embryo is living normal. Okay. But when you will shift the temperature, let's say fly grows at 25 degrees Celsius, normal. You shift the temperature to 29 degrees Celsius, your mutation will not show effect on normal temperature. But as soon as you will shift the temperature, your mutation will show effect. Okay, you will see a phenotype. It will be either dead, okay, organism will be dead, or you will see a specific phenotype. Your organism mutant is viable. It's alive on normal temperature. Now, what you do, you say, okay, I'm going to see whether it plays a role in early embryonic development. You will raise the temperature during early development. If it starts dying, you say, okay, my gene, you have 5,000 such mutants. You say, okay, mutant 1, mutant 10, mutant 13, mutant number 18, mutant number 100. When I bring them to restrictive temperature, which is 29, at early embryonic development, they start dying. They showed lethality. It means these genes are involved in early embryonic development. Same thing you do with the cell cycle mutation. All of them are alive. The people who did this cell cycle screen, all of them were living normal. In the yeast, this screen was done. When they moved this yeast to restricted temperatures at different stages, what they see, okay, this particular mutant 1 is defective in G1 to S transition. Another mutation 2 is defective in S to G2 transition. Then there are mitosis. So that's how these cycle independent kinases and cyclins were discovered. Okay. So these are all tools which you have at your hand. What is time now and how long we have class? We started at 6:15.
Sir, it is 6:54.
Yes, 6:54. Yeah, we'll finish at 7. So now this is the assignment I gave you already. So I expect you have done this. If your mutation is lethal, if your mutation is embryonic lethal, and you don't have temperature sensitive mutations, because these are tricky to generate, is it possible that you can study this particular gene, which is when mutant is showing lethality or death at embryonic stage? Can I study effect of minus minus, which means mutation effect, null effect, at later development? In fruit flies, it's beautiful system, which is the flip FRT system we use. And in flip FRT, since this was part of assignment, I'm not going to explain you. You can maintain your mutant as a heterozygous over a balancer. I gave you assignment for balancers as well. And then using the flip FRT system, you can generate clones. These are the flip FRT. The ones who have done the assignment, they can very easily know what is happening here. You can study loss of function, complete loss of function of this gene at larval stage. This is embryonic lethal when homozygous. Look, you maintain your mutant generation after generation as heterozygous over balancer. But playing with this flip FRT, so flip is recombinase, FRT are the sites where flip is X, and you can generate this, what we call mosaics or somatic clones. This involves mitotic recombination. And then during growth of the tissue, you will generate mosaic, means you have a tissue which will contain wild type cells. You had heterozygous organism. How can it has wild type? Which will have minus minus, and it will have heterozygous as well cells. And you can see the effect of this null cells when they will keep dividing mitotically. You will see a phenotype. So we finish here, and next time we will talk about tricks in mouse. I'll say when you go back today in your free times, try to read about Krelok system. Krelok system is very successfully used to generate through homologous recombination knockout. You must have heard about the word knockout. Knockout means complete gene or a DNA region is removed. So what is Krelok system? You have to read it yourself. Any questions?
Sir, the
Yeah.
assignments you mentioned, when are we supposed to submit them? And are we supposed to do it over LMS or email?
No, you send us, yeah, you can submit it on the LMS as well. But if you go and see my outline, I had given you how to name your assignment, and then also always send to me as well as Ahmad, both. Okay. If you submit it on LMS, still inform us via email. You can simply convert your assignment to PDF and send us to through email as well. Whatever way you find it easy, do it. And yeah, you asked the date for this assignment, which I said, how do you, characterize, how do you discover this? Take your time. You have 2 weeks for this assignment. You have to brush up your classical genetics concept to do this, but do it well. So today is like, I think 18th. Let's say 2 weeks are 1st of March. Take your time. There's no rush. Any other question?
Yes, sir.
Any other question? If there are no more questions, thank you very much. And I wish you all a wonderful evening. Allah Hafiz.